Inhaling and exhaling is the most visible sign of life, and it is frequently mistaken for the entirety of respiration. Beneath that mechanical movement lies the real work, a hidden, microscopic chemical engine running simultaneously inside trillions of individual cells. The sole biological purpose of this cellular engine is to extract usable, life-sustaining energy from the food we digest. The primary raw material for this process is glucose, a tightly bonded 6-carbon molecule delivered to our cells by the digestive system. Respiration is the precise chemical process of breaking those molecular bonds to release the exact energy required for survival. The energy extraction process begins in the cytoplasm, the fluid that fills the interior of the cell. Look at this molecular breakdown. The 6-carbon glucose molecule is too large to process all at once, so the cell actively splits it into 2 smaller 3-carbon chains. These new chains are called pyruvate. Pyruvate acts as the critical biological juncture in the energy cycle. This initial fracture occurs entirely independently of the external environment. It does not require a single molecule of oxygen to proceed. While breaking these initial bonds does release a small amount of energy, the yield is incredibly minimal. Splitting glucose in the cytoplasm is merely a primer. The pyruvate now sits at a biochemical fork in the road, and the path it takes depends entirely on whether oxygen is present in the cell. When a cell is completely starved of oxygen, it is forced to take an emergency biological route to process the pyruvate. Single-celled organisms like yeast regularly face this exact constraint, processing pyruvate through an incomplete oxidation pathway. As this diagram shows, without oxygen to fully break down the molecule, the yeast pathway yields ethanol and carbon dioxide, alongside a very small amount of energy. Human cells can also find themselves starved of oxygen. During intense, sudden physical exertion, the demand for cellular energy outpaces the supply of oxygen reaching the muscle tissue. Instead of producing ethanol, oxygen-starved human muscle cells convert the pyruvate into a different 3-carbon molecule, lactic acid. The rapid buildup of this lactic acid within the tissue is the exact chemical trigger that causes physical muscle fatigue and cramping. This anaerobic pathway prevents immediate cellular death by providing a rapid trickle of energy. However, it leaves far too much potential energy locked inside the chemical bonds to power complex human anatomy over the long term. When abundant oxygen is actively present in the cellular environment, the biological calculus changes. As highlighted in this text, the pyruvate leaves the cytoplasm and enters a specialized enclosed organelle, the mitochondrion. This is the exclusive site for true aerobic respiration. Inside the mitochondrion, pyruvate undergoes complete oxidation. Oxygen acts as a catalyst, tearing apart carbon structures into simple waste, carbon dioxide and water. With bonds shattered, the reaction outputs its true goal, massive generation of ATP, the energy currency fueling all cellular activity. The energy advantage here is staggering. Complete aerobic oxidation produces an ATP yield that absolutely dwarfs the meager output of the anaerobic route. Complex, high-functioning animal life requires this exact scale of energy production. And because this mitochondrial engine requires oxygen to run, our biology is built entirely around an absolute mandate for continuous oxygen delivery. This creates a massive logistical problem. Trillions of individual cells require a constant influx of oxygen, yet the vast majority of them are sealed deep inside a solid mass of tissue, isolated from the atmosphere. The human respiratory system functions as a macro-mechanical supply chain, engineered specifically to solve this logistical challenge. The process begins with a mechanical vacuum. The diaphragm, a broad muscle beneath the lungs, flattens to expand the chest cavity, lowering internal pressure and forcibly sucking air into the body. Once inside, the atmospheric air races rapidly down a branching network of biological piping. The primary tubes divide into narrower bronchi and then further into microscopic bronchioles, systematically distributing the air deeply into every quadrant of lung tissue. The macroscopic anatomy we associate with breathing is a bulk transport pump designed to drag the external atmosphere deep into our internal biology. The air eventually reaches a dead end. millions of microscopic balloon-like sacs at the end of the bronchioles, known as alveoli. This is the location where the actual exchange of gases takes place. Millions of alveoli unfold, creating 80 square meters of surface area for gas exchange. Wrapped tightly in blood capillaries, their exceptionally thin walls allow atmospheric oxygen to diffuse directly into the bloodstream. Simultaneously, carbon dioxide diffuses in the opposite direction, back into the alveoli. This delicate microscopic membrane acts as the precise physiological border where the external environment chemically merges with the circulatory system. Once in the bloodstream, the oxygen requires a specialized transport vehicle to carry it through the body's fluid, the red blood cell. Inside each red blood cell is a protein called hemoglobin, a respiratory pigment specialized to chemically bind tightly to oxygen molecules. The heart acts as a central pressure pump, providing the force required to drive this oxygen-rich blood away from the lungs and into the systemic network. The arterial network carries this oxygen payload out of the chest, pushing it deep into the body's tissues until it reaches every individual waiting cell. The red blood cell delivers the oxygen across the cell membrane, allowing the mitochondria to complete their cycle and trigger a burst of usable energy. Every anatomical component, the expanding lungs, the beating heart, and the rushing blood, serves the needs of the microscopic mitochondria. We breathe for the sole purpose of providing the oxygen required to unlock the chemical energy stored within our food.